So we've looked at the mechanisms that are involved with mitotic division in eukaryotic cells, and we've also had a bit of an overview of the eukaryotic cell cycle and the place of mitosis within that cell cycle. In this video, what we want to focus on is the regulation of growth and cell division in eukaryotic cells. And this quote by Dr. Andrew Murray from Harvard really sums it up very nicely. You know, in a normal situation, this quote from Dr. Andrew Murray from Harvard University sums it up really nicely. In a multicellular organism like an animal, under normal circumstances, normal cells obey strict rules. They divide only when told, and they would rather die than misbehave. And of course, what this is referring to is the fact that under normal circumstances, eukaryotic cells in a multicellular organism have molecular mechanisms in place that will actually cause them to undergo programmed cell death if they are behaving abnormally and in such a way that they could possibly pose a danger to the organism as a whole. And we talked about that before in our cell signaling discussion. That mechanism is apoptosis. This really is a nice summary of the normal behavior of cells, but what we want to understand is how is that accomplished? So what are the sorts of growth control regulations and cell cycle regulations that are in play that make it so that cells under normal circumstances behave like good citizens and don't overgrow or divide out of control, thereby placing the organism as a whole in danger? So first we're going to talk about some growth controls that are in place in the context of multicellular organisms in particular. And these two growth controls that we're going to talk about are contact inhibition, sometimes referred to as density-dependent inhibition, and anchorage dependence. And so if we think about a multicellular organism like us, like an animal, most of our cells are growing in the context of solid tissues, right? So you might have a layer of cells in which you have multiple cells all connected to one another and connected to the rest of the body. So let's say that this is a layer of cells, right? And these cells are connected to one another through proteins in their membranes that form cell-cell junctions. We've talked about these before, so these could be tight junctions, these could be desmosomes, these could be gap junctions. So in a solid tissue layer, the cells are connected to one another, and then also typically they're connected to a connective tissue layer underneath, right? So at the bottom of this tissue layer, connecting this to the rest of the body. And these are also going to be membrane-embedded proteins that are connected up to the internal cytoskeleton, connected up to the extracellular matrix, and form a nice attachment of these cells with one another. So what would happen if there was some sort of an injury that caused the death of some of these cells, or some other event happened that caused some of these cells to die, and we ended up with a gap in our tissue layer where no cells are present? All right, so we have this gap. Under normal circumstances, what's going to happen is that there's going to be growth factor signaling, and those growth factors could be secreted from these surrounding cells. There could be cells underneath that are secreting growth factors in this region. But basically what's going to happen is that there's going to be a growth factor cell signal pathway that's activated that leads to cell division, right? And so under normal circumstances, what we would see is cell division that produces new cells sufficient to fill in that gap that was formed. And then once that gap is filled in and these cells form their attachments to one another and to the connective tissue layer underneath, cell division is going to stop. That is, once these cells make contact with other cells, then cell division ceases. And this is what's referred to as contact inhibition. Making contact inhibits further cell division. So under normal circumstances, what we don't see is cells continuing to pile up and pile up and pile up and overgrow and overgrow and overgrow, right? We don't see that. Now, what's at play here is that these proteins that are making these connections are actually capable of initiating cell signaling pathways to these cells that inhibit further rounds of cell division. So those signal transduction pathways that we talked about in the cellular communication unit, sometimes the receptors or the initiators of those cell signaling pathways can actually be attachment proteins in the membrane. And this is one example of that. So once these cells are connected up and attached to their basement membrane or to cells on either side, we see these inhibitory pathways in the cell initiated that inhibit further cell division. Okay, so that's contact inhibition, or sometimes referred to as density-dependent inhibition. As the density of cells increases, further cell division is inhibited. The second general growth control that's in play in a multicellular organism is referred to as anchorage dependence. So again, we'll start with our solid tissue layer, a bunch of cells here all in a row. These little circles, by the way, are supposed to represent nuclei so that you can tell that these are cells. Imagine that this is, I don't know, let's say a layer of cells in liver, all right? Would it be a good idea if these cells in liver could just sort of break free from their tissue layer and decide, I think I'm going to go and hang out in the lung today? So would it be a good idea if we had liver cells breaking free and heading off into the bloodstream or into the brain, or parts unknown? Obviously that would be a really bad idea, right? In a multicellular organism, your cells and tissues and organs and organ systems have to maintain their structural integrity. They have to maintain their structural interactions and interfaces with all of the other tissues and organ systems in the body. Events like this would really muck that up if they were allowed to occur. So under normal circumstances in an animal, this is not allowed to happen. And if a cell does break free of its tissue layer, then what actually happens is that cell will be killed off and will actually go through apoptosis. So programmed cell death will be initiated if cells lose their attachments to other cells or their basement membrane. And this is what's referred to as anchorage dependence. These cells, in order to stay alive, must remain anchored in their solid tissue layer. Well, how is that accomplished? Well, it turns out that just like we saw up here, we had membrane proteins, attachment proteins that were initiating cell signaling pathways in the cell. Same thing is at play here, different membrane proteins involved. But these membrane proteins actually initiate what are referred to as survival signals in the cell. What they're actually doing is inhibiting apoptosis from occurring or initiating. So in other words, as long as these cells are attached to one another and attached to their connective tissue layer underneath, apoptosis is going to be inhibited in those cells by the signaling pathways that are initiated by these attachment proteins. If the cells break free and lose their attachments to one another, then that inhibition of apoptosis is lost, and so apoptosis is sort of the default state, and it gets initiated and this cell is removed from the body. So these are two general mechanisms of controlling growth and making sure that cells are behaving nicely in the context of the organism as a whole. What I want to talk about next are actually specific molecular checkpoints that regulate the progression of a cell through its cell cycle. So this is that eukaryotic cell cycle we looked at before. We have our interphase out here. We have our interphase out here that is comprised of G1, S, and G2. We talked about this before. G1, gap1, S phase is where DNA is synthesized. G2 is that growth phase. And then we have our mitotic phase here comprised of mitosis and cytokinesis. So there are three really important checkpoints that can be thought of as, points of progression where the cell is actually checking to make sure that molecular events have occurred that need to occur before the cell can move on to the next stage of the cell cycle, or just making sure that it's necessary to move on to the next stage of the cell cycle, depending on which checkpoint we're talking about. So the three checkpoints that are really important and that we're going to look at occur here. The first one is at the end of G1, and this is the G1 to S phase checkpoint. The second is the G2 to M or mitosis checkpoint. So this is regulating the progression of a cell from interphase to mitosis to actually start dividing. And then the third checkpoint is sometimes referred to as the mitotic checkpoint. also a lot of times referred to as the spindle assembly checkpoint. And so this checkpoint is regulating the progression of the cell from metaphase to anaphase, that key point in the mitotic phase of the cell cycle. So if we think about it, we could actually logically work out what the cell's probably checking for at each of these checkpoints, right? So just by thinking about what the cell might be asking itself before moving forward. So just by thinking about what's happening in each of those phases, we can figure out what the cell is likely to be checking for, right? So at the G1 to S phase transition, what is the cell asking itself? It's asking itself, is it appropriate for me to divide? In other words, do I have permission to divide? Is it a good idea for me to divide and produce more cells of the same type that I am? So for example, if you're a yeast cell, what are you going to be thinking about at the molecular level? Well, you're going to be thinking, is the environment favorable to expand this population out? So are there plenty of nutrients? Are wastes not building up to an unfavorable level? If you're a multicellular organism like an animal, that permission to divide is going to be a lot more complex. It's going to be the result of growth factor signals that are coming from other tissues, surrounding tissues maybe, that are signaling a particular population of cells to expand out and divide. So growth factor signals would be providing the permission to divide that's required to progress from G1 to S phase, to start preparing for the next round of cell division by replicating DNA. Well, what about during the transition from G2 to M? Well, at the end of G2, what is the cell really interested in making sure has been accomplished? It's interested in making sure that all of the cellular DNA has been replicated and that any DNA mutations that have been detected or any DNA damage that's been detected has been completely repaired. In addition, the cell might be asking itself, am I large enough? Have all of the cellular organelles been duplicated? Am I sufficient size to be able to support 2 new daughter cells, right? So at G2 to M, primarily we're looking at the status of DNA replication and repair. And we're also looking at cell size as indicators of whether the cell is ready to move on to mitosis and actually undergo division. Now, what's happening at the mitotic spindle checkpoint is exactly what we saw illustrated in that nice movie that we watched in the mitosis video, where we saw the kidney epithelial cells undergoing mitotic division. And remember that we saw that one straggler chromosome that wasn't correctly attached up. It was actually only attached to one of the spindle poles, and it got pulled to one side of the cell. And remember that what we saw in that case was that the cell did not proceed from metaphase to anaphase. It actually waited until that straggler chromosome was correctly connected up and was pulled to the metaphase plate and equal tension had been established. And only then did it move on to anaphase, separating those sister chromatids from one another and pulling them to opposite poles of the cell. So what the cell is checking for at this mitotic spindle checkpoint that regulates the progression from metaphase to anaphase is, are all of the sister chromatids correctly attached, right? And by that I mean, has equal tension been established on those sister chromatid kinetochores? So how are these checkpoints actually regulated? Well, as you can imagine, this is going to involve a whole network of cellular proteins and enzymes that are going to be basically the mechanism by which the cell is regulating progression from one stage to the next. And it turns out that some of these checkpoints have molecular mechanisms in common. So different specific proteins, but belonging to the same class. So for example, the G1 to S checkpoint and the G2 to M checkpoint both rely on proteins called cyclin-dependent kinases. Cyclin-dependent kinases, or CDKs. And we've talked about kinases before, and so you guys know what a kinase is. Kinases phosphorylate targets using ATP, right? So transferring a phosphate from ATP to a target molecule. So both the G1 to S and G2 to M checkpoints are regulated by kinases in this class. Now, why are they called cyclin-dependent kinases? Well, the name is kind of self-explanatory. These are kinases that are allosterically regulated by the presence of another protein called a cyclin. And so this CDK, the active enzyme, is actually a complex that consists of 2 proteins, the CDK itself, the kinase, and the regulatory molecule protein, which is the cyclin. So cyclin CDK has to form in this complex before we can see activation becoming likely. Now, because the mechanisms that regulate these are pretty similar to one another, we're not going to learn the details of both of them. We're really going to focus on the G2 to M transition and look at the details of how that occurs. But everything that we talk about is going to be very applicable to the G1 to S transition as well. It's a very, very similar mechanism. And for both mechanisms, what we see is that the presence or absence of cyclin protein alone is not sufficient to activate or inactivate the CDK enzyme. In fact, there are two mechanisms of regulating CDK activity. The first is presence of cyclin, and the second is making sure that a correct phosphorylation pattern has been established on the cyclin-dependent kinase enzyme itself. So, two modes of regulation. First, cyclin protein has to be present, and then the correct phosphorylation pattern has to be established on the CDK enzyme before it's activated. And so this little indentation here is meant to represent the active site of this enzyme complex, right? So only when both of those things have been established do we see active cyclin CDK then capable of phosphorylating its target proteins. Okay. So we're going to look at both of those mechanisms of regulation in more detail, but we're going to start with regulation of CDK activity by cyclin protein presence. Okay, so again, we're focusing on the transition from G2 to mitosis. So this checkpoint right here, where the cell is checking, is DNA replication complete? Has DNA repair been accomplished? Is the cell big enough before moving on and actually dividing up? So what we see sort of diagrammed here is the CDK that controls that progression to mitosis, so what might be referred to as the mitotic CDK, and then the regulatory cyclin that is going to bind to that mitotic CDK, so the mitotic cyclin. And what we see with cyclins, and the reason that they're called cyclins, is that their expression varies throughout the cell cycle. So for example, for this mitotic cyclin, if we were to look at the concentration of cyclin protein through all the stages of the cell cycle, beginning out here in interphase, what we would see is that through G1, the levels of cyclin protein are pretty low, and then beginning in S phase, we actually start to see an increase in the amount of cyclin protein that's present, and that concentration of cyclin protein actually peaks at the beginning of mitosis. And if we compare that to the activity of this complex, the CDK, remember it's the CDK that has the enzymatic activity, what we see is that the activity of the enzyme peaks and corresponds to the peak in concentration of the cyclin protein. So that makes sense, right? If the activity of the enzyme is dependent on the presence of cyclin, then we would expect that its activity would peak at the same time that expression of the cyclin peaks as well, right? But then what we would see if we were still tracking that cyclin protein concentration is that right here in the middle of mitosis, there is a dramatic drop-off in the concentration of cyclin protein, right? And then as the cell exits from mitosis and enters G1 again, we would start to see that increase, finally peaking at mitosis again. So this is why cyclins are called cyclins, because their expression level increases in predictable fashion in particular stages of the cell cycle and then drops off again, increases and then drops off again, and so forth, right? So same thing would be observed for our cyclin dependent kinase that's regulating progression from G1 to S. As I said, we're not going to focus on that particular CDK. We're going to focus on the G2 to M CDK. So we're going to talk more about this in a little bit, how this cyclin gets degraded and what happens in the cell as a result of that. But this is just sort of explaining why cyclins are called cyclins and how when cyclins are present, we see formation of this complex, which, as we said before, is necessary as a first step to activating this enzyme. Now, again, the second thing that's going to be required is the correct phosphorylation pattern being established on this CDK protein before it can actually start phosphorylating its targets. Well, what are the targets of this activated CDK? Once it achieves full activation, that cyclin CDK complex, and I'm just going to add on that phosphate to make sure that everybody's recognizing that not only is the cyclin going to be present, but the CDK has to be correctly phosphorylated. But once that happens, and this is an activated enzyme complex now, there are lots and lots of targets for phosphorylation that this enzyme is going to be modifying, right? The first one is a set of proteins that are responsible for modifying chromatin structure. So these are called condensins, and as their name implies, they are responsible for initiating the condensation or compaction of chromatin that we see happening at the earliest stages of prophase. Another set of target proteins are proteins that are associated with the nuclear envelope and the nuclear lamina. So when these proteins are phosphorylated, that is what is going to trigger the fragmentation of the nuclear envelope. Proteins in the Golgi get phosphorylated, and that's associated with fragmentation of the Golgi complex, which is necessary for cytokinesis to occur. And then phosphorylation of microtubule-associated proteins and centrosome proteins is required in order to facilitate the rearrangement of the microtubule network in the cell to form the mitotic spindle. So as you can see, all of these target proteins that are phosphorylated by CDK once it's activated are proteins that are associated with the onset of prophase, right? All of these events that are being described here are essentially the beginning of mitosis, and the first stage of mitosis is prophase. So how is activation of this cyclin-dependent kinase actually achieved at this G2 to M checkpoint? Well, to understand that, we actually have to take a step back and think about what's happening during S phase. So we've talked about S phase before, and we know that, the biggest thing that's happening during S phase is that the DNA is being copied. S stands for synthesis. Synthesis of DNA is the big thing that's going on during S phase. But in addition, as you can imagine, as the cell is preparing for that next round of cell division, there are going to be dramatic changes in gene expression. So there are going to be a lot of genes whose expression gets ramped up in S phase in preparation for cell division. And one of those genes is the gene encoding the cyclin protein that's associated with onset of mitosis. So an increase in cyclin gene expression is going to lead to an increase in the concentration of cyclin protein. That makes sense, right? And so during S phase, as we saw in that previous graph, we start to see, this dramatic rise in cyclin protein concentrations. Now, in the context of what else is going on in the cell, CDK is already there. Right? Cyclin-dependent kinase is already there in the cell. It's hanging out, but as we know, it's inactive because it requires both the presence of a cyclin protein and a correct phosphorylation pattern in order to be activated. So what happens when cyclin protein begins to build up in the cell? Well, now we see a concentration of cyclin CDK starting to form. But is this going to be an active CDK enzyme? No, because it doesn't have its correct phosphorylation pattern. So even as cyclin protein concentration starts to accumulate, this cyclin CDK complex is still going to be in an inactive state. So what's required is establishing the correct phosphorylation pattern on this cyclin CDK complex in order to achieve full activation. And so that, as you might imagine, is going to require enzymes that regulate the phosphorylation status of CDK. So the first enzyme that we see coming into play here is an enzyme called Wee1. Wee1 is a kinase, and Wee1 is responsible for phosphorylating CDK, but it phosphorylates CDK in the active site. So here's our cyclin CDK complex. Wee1 adds a phosphate group in the active site of the CDK enzyme. And that is actually not the correct phosphorylation pattern. So this still remains inactive, right? This phosphate is sitting in the active site, preventing the CDK enzyme from being able to catalyze its reaction. Then we see the activity of a second kinase called CA kinase, which phosphorylates the CDK enzyme again. So it was already phosphorylated here in the active site. CAK phosphorylates a second time at a distal site. Now, still inactive because it's got that phosphate group still in the active site. But if that phosphate group were removed, then this phosphorylation on the distal site actually would represent the correct phosphorylation pattern to activate this enzyme. And so what type of enzyme have we talked about before in the class that's responsible for dephosphorylating target substrates? Well, that would be a phosphatase, and that's what we see coming into play next. There is an enzyme called CDC25, and this is a phosphatase, and this phosphatase cleaves off the phosphate group that's on the cyclin-dependent kinase active site, leaving only the phosphate on that distal site, and now this is an active CDK complex, right? So now this CDK enzyme can start phosphorylating its targets. And just to remind you, what are those targets? Well, we're going to see phosphorylation of nuclear lamina proteins. We're going to see phosphorylation of condensins. We're going to see phosphorylation of microtubule associated proteins. As well as other cellular organelles, right? So remember that this phosphorylation of the nuclear lamina is going to lead to fragmentation of the nucleus. Condensins being phosphorylated is going to lead to compaction of the DNA. Microtubule-associated proteins getting phosphorylated is going to lead to reorganization of the microtubule network into the spindle complex. So when do all of these events occur? Well, this is occurring at that initial phase of mitosis that we call prophase, right? These events, when they happen, we call prophase. And so this activation of our cyclin CDK complex is happening at that G2 to M transition. When CDC25 phosphatase removes that phosphate group, this enzyme actually takes the cell into mitosis. Okay, so when does it make sense that the CDC25 phosphatase would be activated then? Only when all of the events that are associated with S and G2 have been accomplished, right? So as you can imagine, as long as the cell is still, trying to replicate its DNA, trying to check and make sure that its DNA has been repaired, trying to make sure that it's big enough, the cell would not want this enzyme to be activated. So let's talk about the regulation of this. So it turns out that this enzyme CDC25 is actually inhibited as long as there's ongoing DNA replication. So there's a whole set of proteins that are collectively referred to as checkpoint kinases that are involved with phosphorylating other proteins in the cell to regulate their activity as part of the cell cycle. So CDC25 itself is phosphorylated and inhibited as long as DNA replication is ongoing. So that makes sense, right? We're not going to get an activated CDK taking the cell into mitosis if the cell has not even fully replicated its DNA. But remember that there are other things that the cell wants to check for as well. And to make sure that those things have been accomplished, we're actually going to be relying on this Wee1 kinase over here. Because remember that Wee1 kinase recognizes this CDK enzyme as a substrate, and when that active site is encountered, it actually phosphorylates it. And so even if CDC25 is activated and removing that phosphate group from the active site, if there's Wee1 kinase in the cell that is itself activated, what it's going to be doing is just adding that phosphate group right back on. And so effectively flipping the switch on this CDK enzyme back to the off position, right? So CDC25 activity is associated with activation of cyclin CDK. Wee1 activity is associated with inactivation of cyclin CDK. Well, it turns out that Wee1 is activated in response to ongoing DNA repair. So as long as there's DNA repair that's happening in the cell, Wee1 is going to remain in an active state, and it's still going to be switching this CDK enzyme off. And it's also activated by small cell size. So as long as the cell is too small to undergo division, We1 is going to be switching this CDK back off, preventing the progression from G2 to M. And so in this way, we see two separate pathways of regulation of cyclin CDK activity through phosphorylation. The first is detecting whether DNA replication has been fully accomplished, and that's the regulation of CDC25. And the second is detecting whether DNA repair has been accomplished and whether the cell is large enough to support two new daughter cells. Right? Now, there's another little twist on this that actually is an example of positive feedback. It turns out that another target of the cyclin CDK complex when it gets activated is the CDC25 phosphatase itself. And so when cyclin CDK is activated, it phosphorylates CDC25 in such a way as to make it even more active. And so that's going to dephosphorylate more cyclin CDK, which is going to be interacting with and activating more CDC25, right? And so we get this positive feedback loop such that once the cell gets cyclin CDK complex activated, we get this progression that leads to more and more activation of more and more cyclin CDK and very rapid entry and onset of the events of prophase of mitosis. So this is an example of a positive feedback loop that we talked about earlier in the semester when we were talking about enzyme regulation, right? So events that the cell wants to initiate quickly and get accomplished quickly are very typically associated with this kind of positive feedback, and this is a good example of that occurring. Okay, so now let's take a look at that spindle checkpoint and see how the cell regulates the passage from metaphase to anaphase. So remember that this is in the middle of mitosis, and what the cell is checking for here is that all sister chromatids have been correctly attached. What we're going to see is that in the case of this metaphase to anaphase checkpoint or spindle checkpoint, the regulation is going to be accomplished through protein degradation. So degradation of specific proteins that leads to a change in what's going on in the cell. And this is going to involve a protein complex called the proteasome. The proteasome is a large complex of proteins that acts to degrade other proteins in the cell. But the proteins that are to be degraded actually have to be marked with a little molecular signal and sent to the proteasome. And so that sort of targeted protein degradation is what is actually at play here in the metaphase to anaphase transition. So how does this work? Well, it turns out that it involves proteins here in the kinetochore that are going to actually leave the kinetochore in response to equal tension being established. So when equal tension has been established on both of the sister chromatid kinetochores, that leads to the release of a protein called CDC20 from the kinetochore. All right. Now, CDC20, it turns out, is a protein that allosterically regulates another protein complex called anaphase promoting complex, or APC. Anaphase promoting complex is an enzyme, and the type of enzyme that it is something called a ubiquitin ligase enzyme. Ubiquitin is a small molecule that can be added on to proteins through ligase activity of different enzymes. So anaphase promoting complex is just one of many ubiquitin ligases in the cell. But the thing is that once this ubiquitin tag has been added on to a protein, that targets that protein to the proteasome to be degraded. So that ubiquitin tag is a molecular signal in the cell that this is a protein that needs to be sent for degradation. And so this tag is only added on to specific proteins that are targeted by this anaphase promoting complex. So if we think about it, as we can imagine, at the metaphase to anaphase transition, can we think of a protein that needs to be degraded in order to see that progression from metaphase, where all the chromosomes are lined up, to anaphase, where they're allowed to be separated? Well, of course, we can see that getting rid of these cohesins or degrading these cohesins would be a good way to get that accomplished, right? Because once that happens, then the motor proteins that are on these microtubules would be able to start pulling these chromosomes apart. But it's actually not quite that simple. It's a little bit of an indirect mechanism by which protein degradation leads to cohesin being cleaved. So the enzyme that cleaves cohesin is called separase. So separase cleaves cohesins. And once cohesins are cleaved, sister chromatids are allowed to separate. Separase is allosterically inhibited by a protein called securin. So when securin is bound to separase, separase activity is inhibited and cohesins cannot be cleaved. So what actually gets targeted for ubiquitination, this little UB is just representing a ubiquitin molecule being added on by APC, is this securin protein. So this securin protein gets targeted to the proteasome for degradation, right? And so once securin is gone, separase can go ahead and cleave these cohesins, and then we can see these sister chromatids being pulled in opposite directions. And when we see that happening, we say that that cell is in anaphase. But there's actually a second target of this anaphase promoting complex, because in addition to targeting securin for degradation, it targets mitotic cyclin protein for degradation as well. So remember that cyclin is bound to our CDK, which was phosphorylated and activated. And so when cyclin gets ubiquitinated and it gets these little molecular tags added on, likewise, cyclin is sent to the proteasome for degradation. So once CDK loses its bound cyclin protein, then that's effectively a molecular switch that flips this CDK enzyme back to its inactive state. So what's going to happen in the cell if there's no more CDK around phosphorylating nuclear lamina proteins and microtubule associated proteins and condensin proteins? Well, remember that whenever we have kinase pathways, there's going to be a corresponding phosphatase that can dephosphorylate those targets. And it's the balance of activity between the phosphatase and the kinase that determines whether the target protein is phosphorylated or dephosphorylated. So if we lose our kinase activity, the phosphatases that recognize those same targets are going to dephosphorylate, and I'm just going to represent phosphorylation with that circle P, are going to dephosphorylate the proteins of the nuclear lamina. They're going to dephosphorylate the condensin proteins. They're going to dephosphorylate those microtubule associated proteins, which I'm just abbreviating as MAP. And so what happens if we dephosphorylate the nuclear lamina? Well, the nuclear envelope is going to start reforming around the newly separated DNA. If we dephosphorylate our condensins, the DNA is going to go back to its open extended chromatin conformation. If we dephosphorylate those microtubule associated proteins, we're going to see the microtubule network going back to its more diffuse structure in the cell as opposed to that spindle structure. And when we see those events, we see that the cell is now in telophase, right? Telophase is essentially the reversal of prophase. So anaphase promoting complex does promote the onset of anaphase by causing the cleavage of cohesins by targeting the securin protein for degradation, but it also is responsible for triggering the onset of telophase by inactivating the cyclin CDK complex that was responsible for holding this cell in mitosis. All of these growth control and cell cycle checkpoint mechanisms for regulating cell division and cell growth in the context of an animal are really relevant to our understanding of how cancer develops and how it can spread and become malignant. So as you can imagine, the onset of cancer is going to involve unregulated cell division, so cells dividing out of control. That's going to lead to tumor formation, but not necessarily tumors that could become cancerous in the sense of becoming invasive and spreading to other parts of the body. This could, in theory, be a benign tumor, right? So cells growing out of control. So mutations would have to accumulate in genes and signal transduction pathway proteins that are regulating cell division and how it occurs. Now, after that, you would have to have additional mutations that are going to get rid of that growth control of contact inhibition and lead to, extensive piling up and expansion of that tumor so that it gets larger and larger, and these cells are not inhibited from further cell division once they've established those contacts. So further mutations are going to be required before we can get really expansion of that tumor and invasion of the surrounding healthy tissue. Now, again, this could, in theory, not be so bad if that tumor remains localized to that one region. So in this case, we're looking at a breast cancer tumor, because at that point, it's pretty easy to get rid of it via surgery, via localized radiation therapy that would kill off that tumor in situ where it sits in the body. Where cancer becomes very dangerous is when we lose that anchorage dependence. And so if the cells of the tumor are no longer limited to dividing by their continued contact with other cells in their surrounding area, they can actually break free and, instead of undergoing apoptosis, survive and encounter blood vessels, lymphatic vessels. So these are the circulatory vessels that allow flow of blood and lymphatic fluid throughout the body. And once the tumor cells reach those circulatory vessels, then they can start to circulate throughout the body, and they can actually set up what are called second site tumors in other areas of the body. And once that happens, then that becomes very, very difficult to treat. And that process of spreading from the primary tumor site to setting up second site tumors is referred to as metastasis. And so multiple mutations have to occur, no matter what type of cancer that you're talking about, that are inactivating the multiple and redundant cell cycle checkpoints and cell division controls and further tissue level growth controls that are normally at work in the body to prevent this kind of nasty behavior on the part of cells. And furthermore, as you can imagine, because apoptosis is a normal pathway that is triggered when cells are behaving badly, in order for any of this to occur, we really have to have mutations that are inactivating the normal programmed cell death initiation or onset. And so mutations that affect apoptosis are very frequently associated not only with breast cancer, but lots of other types of cancer in the body.